Muscular System Assignment 1.2. The overview of the process of muscle contraction. So an impulse transmission from our environment. This is where our sense organs stimulating a change in an environment and sending an impulse down the central nervous system, where the central nervous system will send an ongoing impulse for the muscle to contract. The neuromuscular junction is a gap between two nerve endings where information needs to be carried. And then the sliding filament theory is the way the muscle contracts. So there's a number of different stages to allow the actin and myosin to attach to each other for the muscle to contract in the way of shortening and lengthening. And these three different sections all go together for muscle contraction. So the nerve impulse is our sense organs, which are our eyes. The nose is the smell and skin by touching things. This travels along the sensory neuron into the central nervous system, what sends an impulse down the motor neuron, and then this that later gets onto the neuromuscular junction, which is the gap to jump, and this is carrying the information. The salutatory conduction is where these impulses need to happen so quickly. The way they happen so quickly is by doing a domino effect, where the impulse is jumping along the nerves. And as you can see in the picture, how the impulses are jumping along, and this quickens up the process. So the neuromuscular junction is a chemical synapse, and the chemical synapse is this gap in between two nerve endings, where the information which has been carried down the impulse needs to jump this gap for allowing the muscles to understand the type of contraction they need to do. When this jumps the gap, it transmits a signal down the T-tubules, which I'll go into a bit more depth in a minute. And this sends the information down into the myofibrils. And this is basically the start of muscle contraction when it jumps over this neuromuscular junction. So this is a sliding filament theory overview. It's quite a difficult way to understand how muscles contract. So this is just a simple, brief way, and then I'll go into more depth, which each stage means. So you have your stimulus, and that is your nose, smell, and these are all your senses. So in the sporting context, we're going to talk about netball. So I've caught a ball, and then I'm going to pass the ball back. So that is my muscles contracting, passing the ball back. And then when I go on to the muscles that are relaxing, it's when the ball's left my arms and I'm not doing anything else. I'm just waiting for the ball to be passed back again. So when this happens, the central nervous system sends impulse down the motor neuron, which is carrying all the information we need. Then I have to jump over that neuromuscular junction, which is the synapse. This then goes down the T-tubes, and this T-tubes is sending the information. It's a quick way to send the information and basically triggers the sarcoplasmic reticulum, and this is the release of calcium ions. So this information being passed down the T-tube stimulates this release of calcium ions. What I'll explain in a minute what these calcium ions role is, and these go into the myofibrils. And the myofibrils is a part of the muscle. So the general structure of the muscle is you have many muscle fibers, and these are bundles of muscle fibers make up a fascicle. But inside these muscle fibers, there's another layer called the myofibrils, and it's basically very small pieces where these calcium ions are going to, and all this information to be sent to. And these myofibrils contain the actin and myosin, and you need actin and myosin. That's basically the way muscles contract, by them pushing in, and there'll be diagrams of what they look like and what they are. And this basically leads to the muscle contracting. So how the muscle contracts according to the sliding filament theory. So the sarcoplasmic reticulum is where the calcium ions are being released. So the A is actin, and this is the thin protein. So as you can see in that diagram, you have the thin filament and thick filament, and the thin one is the actin, and then the myosin is the thick filament. The myosin has arms, so the process is the myosin stays still, but what happens is the myosin gains energy to attach to the actin and pulls the actin along in towards the M line. So the N lines will go, no, the Z lines will go inwards towards the M line for the muscle to shorten. And then when the muscle's relaxing, the Z lines go out, the actin goes back out. However, it's not so simple. The myosin cannot attach to the actin right now, and this is because it's been covered by troponin and tropomyosin. So this is a diagram of actin. So the myosin wants to attach to these actin binding sites. However, the actin binding site is being blocked by the tropomyosin strand, and that's the orange strand, and the troponin is covering the tropomyosin. So the role of the calcium ions, it's the calcium ions do a reaction with the troponin and the tropomyosin, and actually pulls these away from the active binding sites and changes the whole shape, which allows the actin binding sites to be visible for the myosin to attach to. So it's the same diagram where, let's get this all up. So you have the sarcoplasmic reticulum, which is releasing the calcium ions. The myosin, which does not move, needs to so the calcium is attached to the actin, and this is released all the troponin and tropomyosin, so the actin is visible for the myosin to attach. However, the myosin needs to have energy, so it takes ATP. And ATP is basically a sugar substance which gives it the energy to attach to the actin. So this goes to ATP, and pulls the actin in towards the M line, and when it pulls in towards, the Z lines go inwards as well. And this is forming a cross bridge where the actin and myosin are attached. However, then the actin needs to be released from the myosin, so it takes another bit of ATP, which energizes the myosin to release and attach to another one. So this is muscle contraction. As you can see, the Z lines moved in towards the M lines, and the muscle has shortened. And this red rectangular shape is the myofibril, and as you see, it has shortened and got smaller. And then this is back at rest, where it's normal size. The actin is moved out, so the myofibril is lengthened again. You also have the sarcomere, and the sarcomere is basically these actin and myosin. There's blocks of it all the way down the myofibrils and myofibers. And these sarcomeres, when the muscles contract, they get smaller with it, and then they expand. So as you can see, there's different sections of this all down the myofibril. And this is at rest. So at muscles at rest, it's basically the total opposite of contraction. So there's no stimulation from the central nervous system. So there's been no change in the environment for your muscles to need to contract. So the sarcomere is at rest, so this is normal length. So the Z lines are not any closer together or any further away. There's no cross bridge, so there's no connection between actin and myosin. However, the myosin is still energized by the ACP. But as the actin is being recovered by the troponin and tropomyosin, it's not attaching to make that cross bridge. So when muscles contract, there's 4 different types of contraction: isotonic concentric, isotonic eccentric, isometric, and isokinetic. Isotonic is when the muscles change length and it contracts and causes movement. Isotonic concentric contraction is where the muscles shorten when the muscle contracts. It still generates force and it overcomes resistance. For example, weightlifters. When they're lifting the weight towards the body in horizontal flexion, the bicep brachii is shortening and getting smaller, and that is concentric contraction. And the opposite is isotonic eccentric contraction, is where the ends of the muscles are lengthening. So again, you could do the example of weightlifters when they're putting the weights back down. The bicep brachii is lengthening and the tricep brachii is shortening. Or you have another example is like kicking a football, where you extend your leg to kick out and your hamstring muscles are lengthening. These movements can be voluntary or involuntary. Like your involuntary movements would be reflexes where you're not actually thinking about it. Isometric contraction is where both ends of the muscles stay the same length and strength, and there's no movement at a joint. So this could be like holding a press up, where your joints are moving or they're staying in the same shape and your muscles are not lengthening or shortening. However, your muscles are still contracting because they are still working, but just are not moving. So this is holding a press up or pushing against the wall, or maybe holding a tennis racket because your muscles are squeezing the tennis racket but are not moving. Isokinetic contraction is where the muscles shortens at constant rate of speed. And also very good at targeting specific muscle groups. They're very rare, so an example would be breaststroke in swimming, is where the water provides a constant even resistance to the movement of adduction, so the muscle is contracting at the same rate of speed with the same amount of force each time.